Instructions for use of nucleic acid extraction kit and lung cancer mutant genes detection panel. Display of nucleic acid extraction kit. Show the contents. Instruction for use: DNA, RNA spin columns, collection tubes, centrifugal tubes, and other various reagents. Buffer FB, buffer DW1 and 2, buffer RTW, buffer TC, and so on. Note: for the first time use, add 21 ml ethanol, 96 to 100% into buffer RTW. Add 17 ml ethanol into buffer DW1 and mix thoroughly. Add 24 ml ethanol into buffer DW2 and mix thoroughly. Tick a checkbox on the bottle label. Check to ensure that all reagents have been added with alcohol. Sample pretreatment. Check sample. Add 700 liters buffer FB to 20 to 60 mg tissue samples. Homogenize by grinding. Transfer 600 liters tissue homogenate to a clean 2 ml centrifugal tube. Add 20 L proteinase K solution into the tube above. Mix gently by pipetting up and down. Vortex mixing. Incubate at 56 degrees centigrade for 15 minutes. Centrifuge at 14,000 revolutions per minute for 3 min. Transfer the supernatant to a clean 1.5 ml centrifugal tube. For RNA extraction, the remaining precipitates are used for DNA extraction. DNA extraction: add 180 liters buffer DTL into the underlying precipitates. Mix by vortexing. Add 20升 proteinase K solution. 56 degrees centigrade for 30 minutes for lyse the sample. Add 200 liters buffer DTB and 200 liters ethanol. Mix by vortexing. Take out the DNA spin columns and collection tubes to milliliters. Transfer the entire lysate to the DNA spin column. In the 2 ml collection tube, without wetting the rim. Centrifuge at 10,000 revolutions per minute for 1 min. Discard the flow-through in collection tube. Add 600 liters buffer DW1 to DNA spin column. Centrifuge at 10,000 revolutions per minute for 1 minute. Add 600 liters buffer DW2 to DNA spin column. Centrifuge at 10,000 revolutions per minute for 1 minute. Discard the collection tube with flow-through. Place the DNA spin column in a clean 2 ml collection tube. Centrifuge at 12,000 revolutions per minute for 3 minutes. Discard the collection tube with flow-through. Place the DNA spin column in a clean 1.5 ml centrifugal tube. Add 30 to 100 liters buffer DTE to the center of the membrane. Incubate at room temperature 15 to 25 degrees centigrade for 1 to 5 minutes. Centrifuge at 12,000 revolutions per minute for 1 min. The eluted DNA is immediately ready for use or for storage under -20 degrees centigrade. Add 320 liters ethanol, 96 to 100% into the supernatant and mix. Take out the RNA spin columns and collection tubes to milliliters. Transfer 600 liters lysate to the RNA spin column without wetting the rim. Close the lid and centrifuge at 12,000 revolutions per minute for 30 seconds. Discard the flow-through in collection tube. Add 600 liters buffer LTW2 RNA spin column. Centrifuge at 12,000 revolutions per minute for 30 seconds. The washing step using buffer LTW should be repeated once. Discard collection tube with flow-through. Place the RNA spin column in a clean 2 ml collection tube. C. Centrifuge at 12,000 revolutions per minute for 5 minutes. Place the RNA spin column in a clean 1.5 ml centrifugal tube. Add 30 to 80 liters buffer TE to the center of the membrane. Stand at room temperature, 15 to 25 degrees centigrade, for 1 to 3 minutes. Centrifuge at 12,000 revolutions per minute for 1 min. The eluted RNA is ready for use. If it's not used immediately, it should be stored at -80 degrees centigrade. Lung cancer mutant genes detection panel. Retrieve all components from the reagent kit, including the instruction manual, test strips, enzyme mix, reaction solution, and positive control. Take a test strip and mark it properly. Each PCR run must contain positive control and negative control. In the following 3 tubes, add the sample, positive control, and negative control. Add the enzyme mixture respectively to each tube. Add 5.3 micro-L solution into strip DAH, and cut the PCR tubes. Add 6.2 micro-L solution into strip RAH, and cut the PCR tubes. Briefly centrifuge the PCR tubes to collect all liquid at the bottom of each PCR tube. Use OB 7500 for testing. Enter experiment name. Please set up as follows: reporter die, FAM, VIC, ROX. Quencher die, none. Passive reference, none. Assign targets and samples. Set up the PCR protocol using the cycling parameters. Start the PCR run immediately.